All right, good evening, everybody. I apologize for my raspy voice. It's the first day in a week I've actually had a voice. So I'm happy to be here with you this evening. I gave a public presentation last week and lost my voice right after. Anyway, welcome to the Coastal Studies Institute. I'm Reed Corbett, the Executive Director here at CSI. And we're excited to continue our series of Science on the Sound. So this is the first of 2026. We've got several lined up. I encourage you to, keep the eye out for John's emails, for CSI's emails. You can sign up for our newsletter and other mailings that we send out announcing the different events that we have. There's a sign-up sheet right out there. Feel free to take any swag or pamphlets that you like as well. I'll also mention that we do, for you or for people that you know, we do guided tours here at CSI the second and fourth Wednesday of each month at 10 a.m. And we do have a lifelong learning program coming up on January 30th, which is a waterfowl birding trip to Lake Mattamuskeet. So again, keep an eye on our socials. John keeps posting things of all the things going on, so thank you. We're also excited. We've got several of our undergraduates here. We've got a new cohort of undergraduates from ECU spending this semester with us this spring, so glad y'all could come out. Tonight, we're happy to welcome Dr. Jim Morley. Jim Morley is an assistant professor in the Department of Biology and a scientist here at the Coastal Studies Institute. Jim has a degree in biology from SUNY Cortland, and then 2 degrees from NC State, which is all right, I guess. No, they're a great partner of ours. Jim is a fisheries ecologist, been investigating the life history of marine and estuarine species in North Carolina for over 20 years. He's interested in how human-caused disturbances and climate change interact with the life cycles of aquatic species, and we'll hear some about that tonight. So thank you, Jim.
Thanks. Thank you all for coming and braving the cold and the dark, and thanks for the introduction, Reed. I lead the Marine Fisheries Ecology Lab here at CSI, and we have a pretty broad research focus. One of the things that we try to do in our research is try to learn a little bit more about the life history of marine species, and kind of that element of our work is what I'm going to be focusing on today, and specifically with our toothiest fish. So Sheepshead really have earned the name as one of North Carolina's toothiest fish. Many of you are probably familiar with Sheepshead, but they have these, they have this mean set of incisors that are very, kind of human-like, and then they have an impressive set of pharyngeal teeth, both on the floor of their mouth and the roof of their mouth. And so these pharyngeal teeth aren't unique to Sheepshead. Lots of different species of fish have those, but Sheepshead kind of kick it up a notch a little bit. And so they have these teeth to, based on what their diet is. So Sheepshead have really one of the most diverse diets in the fish kingdom, really globally. They basically eat everything that's not a fish. There might be some oddball occurrences where they've eaten a fish, but they basically just graze everything in sight that's an invertebrate. Crabs, I guess I gotta get used to using these things. All right, so, sorry, there we go. Crabs and hydroids and shellfish and, various invertebrates that you might associate with like biofouling communities. So, if you're resurrected as a hermit crab, this is probably gonna be the last thing that you ever see. So, before I talk about my Sheepshead journey, I'm going to go rewind back in time about 20 years to when I was a graduate student, as Reed said, when I was at NC State. At that time, I was doing research on bluefish, and I lived down in the Morehead City area. So bluefish are another very toothy fish. I like toothy fishes. And I'm not going to go in depth on that particular project, but that project involved doing a lot of bottom trawl sampling in these 2 particular areas, by Cape Lookout and then by Cape Fear. Those were kind of our sentinel sites, and we went back and sampled there monthly for many years. And so Most of you living in this area are familiar with what a bottom trawl is, but just in case, because this will come up a number of times. So bottom trawl sampling is just towing a mesh bag across the seafloor. You're probably used to hearing about bottom trawls in the context of fishing, but it's also really valuable scientific sampling gear because it is good at catching, organisms on the seafloor. So this is actually a picture of me 20-some years ago out on a shrimp trawler processing a fish catch for bluefish. So anyways, back to this. We did a lot of sampling in these regions, and once in a while in the spring, we would get these huge catches of sheepshead, in the neighborhood of Cape Lookout. And so catching sheepshead was pretty rare in this habitat to begin with, because we're kind of sampling in sandy, muddy bottom. We're avoiding rocks and things, which is what you typically think sheepshead like. So, it was really conspicuous, and we thought potentially these, we just happened upon a spawning aggregation of sheepshead. Lots of species of fish form up into big groups to spawn and reproduce, and then they'll disperse. So, when we saw a catch like this of sheepshead, we were thinking, well, maybe there must be thousands of them down there, right? But at the time, I didn't have much time to think about sheepshead. I just kind of filed away this information in my back pocket for a later day. So a little bit of background on the life history of sheepshead before we kind of dig into what my research has been. Sheepshead are very structure-oriented species throughout their entire life cycle. The adults, where people are trying to fish for sheepshead, they're really well known to aggregate around bridges and pilings and port walls and any kind of rocky structure. They also like oyster farms. They also like seagrass beds. Even the big adults like seagrass beds. And the juveniles also like structured habitats, especially seagrass beds. Juveniles, as far as we know, are completely estuarine dependent. There's no evidence of the really small juveniles using the ocean habitat. They all seem to come into the estuary first. Recreational interest of sheepshead has increased quite a bit. So if you look at the graph there, landings of sheepshead, this is recreational landings. Most landings of sheepshead are recreational. Have fluctuated over time, but since 2017 or so, they've been pretty high. And then if you look at that gray line, that's the number of directed trips where people are actually going out to fish for sheepshead. That has increased. So there's really has been a pretty big increase in interest in sheepshead. And I think if you're involved in recreational fishing, you've probably noticed that. There's currently no stock assessment for sheepshead. They're one of the most recent additions to the state fishery, to the list of species that the state's responsible to manage. But there's currently no stock assessment, and there's no fishery management plan. Those things are in the works, though, I should say. And there's critical knowledge gaps in sheepshead around, their reproductive life cycle. There's critical knowledge gaps around the early life stages of the species, and just their movements. How far are they moving around? How are they using habitat? And so, after I was done with my graduate work, I found myself about 10 years later, I was a postdoc at UNC. So I went from NC State to UNC, and now I'm at ECU, so I'm kind of doing the rounds. I had the opportunity to do some research on sheepshead. We got some funding to do research on sheepshead. I had this information kind of tucked in my back pocket of where I thought sheepshead might spawn. And so we decided to try to go out and try to catch sheepshead in the act. Go out, catch them, and verify that they're actually spawning in some of these locations. So that's the first part of what I'm going to talk about today. There's going to be 3 parts today. So specifically, we decided to look for spawning sheepshead in this area around Cape Lookout. And so what we did was, this took place back in 2018, 2019. It was generally known that sheepshead are spawning in the spring, so we went out during April and May. And all these marks on the map here, don't worry about the different colors there. I'll get to that in a few slides. But those were all the locations where we sampled for sheepshead to see if we could catch spawning sheepshead. And so just to give you an idea of what that looks like when we're going out and sampling for sheepshead with this gear, I've got a couple of videos. Just trying to highlight the biodiversity of things that we catch out there. So do you all know what kind of shark this is? Anybody know what kind of shark this is? What's that? Right, it's a sand tiger shark. Another toothy fish. Only toothy fish today. We catch, we're out here sampling in the sandy, muddy bottom, but there's some really interesting biodiversity in those areas, 1 to 10 miles off the shore. So a lot of people are fishing on the wrecks, but there's a lot of interesting species in between the structure. So on the left is a picture of a sand tiger, and that's a thresher shark right next to it. A lot of stingrays. There's a juvenile cobia. And so, we're out there to catch sheepshead. We try to minimize injuring and killing other species. So our basic protocol is the fish hit the deck, we measure and count all of the sharks and rays and throw them back immediately. And they actually do very well, especially during these cold months. And then we try to minimize the catch of the smaller fish by using a trawl that has really big mesh size. So we're just trying to catch big sheepshead, so we use a really big mesh size. So the vast majority of small fish just swim right through the net. And then another video, this was a video where we were successful. So here, this was off of Core Banks. So here we caught a lot of sheepshead. So we were happy that we went all that way and actually caught sheepshead. The fish in the foreground there is called an angel shark. It's kind of a hybrid between a shark and a stingray, so another interesting species. So again, we went out to all these places around Cook Cape Lookout to catch big sheepshead, hopefully catching them in the act of spawning, and we were successful. We caught big sheepshead, and on the right there, we even brought home dinner. For those, that's what we do with the male sheepshead. We really, for this reproductive studies, we're mainly interested in the females. So males either get thrown back over or they get eaten. Okay, so back to this map here. These symbols here are the locations on the map where, again, where we sampled. The blue dots are showing areas where we caught female sheepshead. The red X's, we either didn't catch any sheepshead or we might have caught just some males there and no females at all. So we caught these sheepshead in these areas during the spawning season. We had to bring them back to the lab to verify that they're spawning. So we take the gonads out of the females, and we also take the otoliths, what are called otoliths, they're ear stones from the fish's brain case, basically. And we can use those otoliths to age the fish. You can count rings on otoliths like rings on a tree to determine how old the fish was. So this is getting into some of the results of that. So we shipped off samples of gonad out to a histology lab, and they take a cross-section of the gonads, and they use some specialized stains that allow us to see what's going on in there. So what you're seeing in here are sheepshead ovaries. So each one of these cells is an oocyte, so it's a developing egg that will eventually be spawned by this individual fish. So, and again, it's very colorful because of the stains. So first of all, by looking at these slides of sheepshead gonads, we know immediately that sheepshead are what we call batch spawners, meaning they don't spawn all their eggs at once and then just they're done for the year. They have a spawning season that might last 1 or more months, where any given female is going to spawn some one day and then maybe wait a couple days and then spawn again for some period of time. And we know that just because we see all these different oocytes at different stages of development. Some are ready to go, some might be ready in a couple of weeks. We found that every single female we caught, they were all in a condition that we would call an imminent spawner. So every one of them had really advanced oocytes that were ready to be spawned probably within the next 12 hours. So this was kind of telling. Every female we caught was about to spawn. So that really suggests this is a really important spawning habitat. This is actually a hydrated oocyte. The HO stands for hydrated oocyte. They're the oocytes that are really big. They're right about to be ovulated and then spawned within hours. They look like amoebas here because during the staining process, they get all wilted up and they end up looking like that. But naturally, they're the big ones that are ready to be ovulated and then spawned. Another interesting thing we saw from these slides was every single female had post-ovulatory follicles, which we call POFs. I mean, this is very similar to human biology, actually, if you remember back to your high school biology. But these things here are the POFs. That's what's left over after the fish ovulates the hydrated oocytes and then spawns. So if we see those POFs, that tells us that that individual fish has spawned within the last few days. So that was kind of neat. Every female we found was about to spawn, and every female we found had probably spawned within the last few days. So it shows that they probably have a pretty high frequency of spawning during the spawning season. So in addition to looking at that, we were able to estimate the fecundity of sheepshead, so how many eggs do they release? And we did this, I won't go into too many details, but we basically take a sample of the gonad. Here's a zoomed in portion of the gonad. You can see all the different oocytes. We disperse them on a Petri dish, and then here's a microscope view of that. So you can see the big hydrated oocytes, the HOs, and then you can see the earlier stages, the smaller oocytes. So we're basically able to count all of the hydrated oocytes and then extrapolate back to the full size of the gonad, and we can tell how many eggs are going to be released. So the sheepshead were all aged. Our colleagues at DMF aged the fish for us. They were all from between 7 and 22 years old. Some sheepshead off North Carolina have been aged to be over 30 years old, and I think in Virginia even over 40 years old. In talking a little bit about this fecundity, so this graph is showing, each one of these dots on the graphs is an individual fish, and it's telling you what the fecundity of that fish was. So this fish over here, its fecundity was about 120,000. So this is in thousands. I got to use my tool. So this fish, it's not as easy as it looks. This fish here had a fecundity of 120,000. And a lot of things to remember. So the average fecundity of all these fish was 264,000 eggs. So every time one of these female sheepshead spawns, they're releasing over 200,000 eggs. And then again, they might do it again in a few days. So very, very high fecundity. And again, that's what we call this batch fecundity, because they're batch spawners. So that's how many eggs are released during each spawning event. We also found that fecundity increases disproportionately with size. And I won't get into the functions that we fit there, but that's how we've determined that. And so basically, as a fish grows, its reproductive output even grows faster than its body size. So if you take a sheepshead, if it doubles in size, its fecundity is more than going to double. So it's like a disproportionate increase. And so that's really important from a fisheries management perspective, because if you have two hypothetical populations of a species, so say this group on the left consists of more fish, but the fish are smaller spawners, and this group on the right consists of fewer fish, but they're bigger, and the overall biomass is the same between these two hypothetical groups, the bigger fish are still going to have a greater reproductive output. And so we call those in fisheries ecology BOFs, which stands for big, old, fat, fecund female. And so that's one of the reasons why we have slot limits for things like red drum. If you conserve some really big old fish in the population, the thought is that they have a higher contribution of reproductive output than smaller fish. All right, part two. So we found these spawning fish off of Cape Lookout. And again, every single female we caught appeared to be about to spawn, and they're actively spawning out there. So we wanted to determine how widespread these spawning aggregations are in the southeast US. And the way I did this was basically just to scour publicly available data from scientific agencies. In particular, I looked at data from this sampling program called CMAP, South Atlantic, that's conducted by the South Carolina Department of Natural Resources. So that group samples in this same habitat that we were sampling every year in the spring and the summer and the fall. And that's what all these X's are along the coast. These are the locations where CMAP samples, and they use that similar type of bottom trawl gear. So they do it because their data goes into helping manage fisheries, so they can assess the health of populations. And then I also pulled together some data from my graduate work and included the data from where we had just sampled and caught those fish. And what I'm looking for is just any individual trawl that had a big sheepshead catch, because again, it's kind of unusual to catch sheepshead in this habitat. So if there's a big catch, then that might suggest that the trawl pulled, or, the trawl went through a spawning aggregation. And so now I'm going to focus into this area here where the red box is. So amidst all that, trawl data that I sifted through, I didn't expect to find many big catches of sheepshead because I think they're quite rare, and the boat has to be sampling just at the right time that year just to happen to sweep through because there's some variability there. And I found 17 different occurrences where one of these scientific trawls caught a big catch of sheepshead that would indicate that sheepshead were spawning in that spot. And so each one of these bubbles on the map is indicating one of those locations. And so the interesting thing with this is we looked at data from Cape Hatteras all the way to Cape Canaveral, Florida, and we only saw these evidence for these aggregations right here off of Cape Lookout up to Cape Hatteras. So that was kind of unusual. And it might suggest that there's something unique about this area that attracts spawning sheepshead, probably the proximity to all the extensive seagrass beds. All of these spawning aggregations were encountered between 17 and 21 degrees Celsius, so that's probably our window of fertility for sheepshead. Every species of fish has a particular range of temperature where they're able to reproduce, and this, we're suggesting this is the temperatures where sheepshead spawn. And we also, all of these aggregations appeared to be between, or they all were, between April 12th and May 17th. So this might indicate the bounds of a spawning season for sheepshead off of North Carolina. And so we were able to test that, that hypothesis that the spawning windows between early April and late May by looking at juveniles, and I'll explain why in a second. So this was kind of, by looking at the juvenile fish, we can further test this hypothesis of when the spawning season is. And so that propagated this new study that we conducted, and this was after I came to ECU. And this was part of Maddie Johnson's master's thesis, and she graduated a couple of years ago. So what she did was go out and collect juvenile sheepshead, and we can collect juvenile sheepshead, we can pull the otoliths out of their brain case again, so I'd already brought up otoliths once, so we can pull out the tiny sheepshead otoliths, we can section them, and basically that means we take a thin deli slice-like slice through the middle of the otolith. And when we do that and we sand it down, polish it, and look at it under microscope, we can actually see daily growth rings on the otolith. So that's what you looked at here. This is the cross-section of the otolith. And these lines are the daily growth rings of the otolith. So all we have to do is count those daily increment rings, and then we know how old the fish is. And because we know when we caught the fish, we know what its birth date was. And so there's a lot of information here, but I'm going to simplify it in a second. So Maddie sampled fish in 3 basic areas, up around the Oregon Inlet area, around the Hatteras Inlet area and the seagrass beds in those areas, Cedar Island Bay, and then down here in Back Sound around the Beaufort area. And the ABC here is corresponding to the ABC in the figures. And so these graphs are showing the distribution of birth dates that we calculated for all the juvenile sheepshead that we caught. And just to summarize that, this shaded area is that spawning window that we hypothesized based on catching the adult fish. So as you can see, the vast majority of the juvenile fish had birth dates within that window of mid-April to late May. So that really does seem to be the core spawning season for sheepshead. We did see some evidence of fish spawning even earlier than that, and we didn't really get out there and sample that early, so that makes sense that we weren't able to detect that they were spawning before then. All right, another time for this? I think so. Another aspect of what we were able to find with looking at the juvenile otoliths is this settlement mark. So if you look at the whole otolith, there's this dark region in the middle, and there's this prominent line here that goes around there. So what that indicates is that's the age the fish went from a larval stage to the juvenile stage. So a little background on fish life cycle. Fish, hatch out of their egg. They spend time as larvae in the plankton, so they're swimming around in the water column, more affected by ocean currents. And then once they get to a certain size, they start to put on their color. They start to look more like a baby version of what they are, a baby sheepshead. And that's when they need to find their nursery habitat. In this case, might be a seagrass bed for a juvenile sheepshead. And so that settlement mark, if we count the rings leading up to that settlement mark, we know how old the fish was when it made this transition to nursery habitat. And so what we found there was that sheepshead have an average settlement age of about 25 to 30 days. So they basically hatch out of their eggs. They spend about a month in the planktonic community, so up in the water column, drifting around with currents. So, which is, as far as fish are concerned, that's a fairly short larval stage. And then after that, they need to find nursery habitat in an estuary. So these are just the different areas where we sampled the juveniles, and you can see the mean here. The mean here is generally between 25 and 30 days. One thing that was kind of interesting is the juvenile fish that we sampled by Oregon Inlet had a significantly longer larval stage. And we think that's probably has to do with those fish are caught above the Hatteras break. And Cape Hatteras is this really substantial break point for a lot of species. So a lot of times species are genetically distinct on either side of Cape Hatteras, and we think that could be evidence for that. All right, moving on to the last part. So sticking with the juvenile fish, we've been very interested in the habitat use of juveniles, and I've talked a little bit about that. But this is a particular study on that. So as a fisheries ecology lab, when we investigate juvenile habitat, or when any marine scientists investigate habitat value, the most common way is to use nets and traps, like here a beach seine, and that gives you a snapshot of what species occur in different types of habitat at any one time. And that's a really good tool to understand the relative value of different habitats for different species. However, it's somewhat limited in that we don't really get an idea for how the fish are using the habitats, how much are they moving around in those habitats, are they using adjacent habitats, what are some of the interesting aspects of the fish behavior in those. And to do that, we did a tagging study, again, with juvenile sheepshead. And so this represents Andrew McMain's PhD student. Andrew's back there, so I'll try not to mess up his research in front of him. But what he did was he tagged 27 one-year-old fish, all about this size, less than 200 millimeters, and he used acoustic telemetry tags. So that's the picture of that's what the person's holding there. It's a tiny little pill-sized transmitter tag, and I'll talk more about that in a minute. But he used veterinary protocols to surgically open up the fish, insert the tag, and stitch the fish back up. So on the bottom right there, you can see the tube going into the fish's mouth. So that keeps water flowing over the fish's gills, and there's anesthesia in there, so the fish is knocked out. And so there's a whole process with this. And we did this down in Cedar Island Bay at an oyster farm there, Jay Styron's oyster farm. So the way this works, just really briefly, this is clearly not a sheepshead. I just found this image on the web. The fish is tagged, and this tag is constantly sending out this ping, this ultrasonic ping that you can't hear with the human ear. And then we have these receivers, these listening devices stationed all around the estuary to pick up those pings. And with those, and understanding the physics of sound and water, we can triangulate the location of the fish and track its movements at a really fine scale. So that's what we did with sheepshead on this oyster farm. The fish seem to do sheepshead are a very hardy fish. Andrew kept some fish in an aquaria. He did the surgery on the fish and put them in an aquaria to see how well they heal. So you can see how they heal. They're looking pretty good at 4 days. And after 2 months, the fish is completely healed. The sutures are completely healed. He even was lucky. He recaptured a couple of fish that he had tagged and let go, and they look like they were healing as well. That's the fish in the middle. All right, so this is a video of maybe the one day I went out and helped Andrew, I think. So is letting the fish go. And so away we go. So look how happy that tagged fish is. And also note, look how the water's really murky in this system, as most, muddy bottom North Carolina estuaries are. So that'll kind of come into play in a minute. So the main thing I'm going to show you here is this kind of cool animation in a second. But before I show you, I just got to kind of tell you what you're going to be looking at, and then you can watch it. So the organization of this, up at the top there, you can see the date stamp and the time stamp. So this is all representing actual data. This animation is based on actual fish movements. This video is going to play through a 24-hour period in fast forward, so it's going to take maybe a minute to get through a 24-hour period, and it's going to start at midnight and cycle all the way through to midnight the next day. Each of those colored dots is an individual fish, so you're going to be able to watch these fish swimming around. I think there's 15 to 18 fish that are in the array at this time. The black polygon is showing the boundaries of the actual oyster farm, so you can see how fish are using the oyster farm. And then that gray box, the bigger gray box, you can kind of ignore that. That's just a drone photo that we pasted on top of a Google Earth photo, so you could see the oyster farm better. All right, are you ready? Okay, so again, we're starting at midnight. It's 1 a.m. It's 2 a.m. You can see all the fish. You can see how they're hanging out using the oyster farm. 5 a.m., 6 a.m. Okay, they woke up. So one thing we learned with this study is that sheepshead sleep. So again, this is actual movements over a 24-hour period. So we just passed noon, 1 o'clock. And so some of the interesting things that we observe with this data are I'll let it play through. So now it's back to evening, 8 o'clock, 10 o'clock, and they went back to sleep.
So 8 o'clock p.m.?
Yes.
Okay.
So it was in the summer, so right. And now it's just starting over again. So again, some of the really interesting things we learned with this are, sheepshead really liked the oyster lease. A lot of them were really hanging out in the oyster lease. And as you watch them, some of them are just going up and down the oyster bag lines, really staying tight to the structure. Another interesting thing is that these juvenile sheepshead are really mobile. Some of them are kind of hanging out on pilings, but others are really constantly on the move. A bunch of them are going off exploring other habitats. There might be some of the marsh edge, and maybe there's some seagrass in there. And those guys are covering a lot of ground pretty fast. And also the sheepshead really seem to be able to navigate in this area. Some of them will zig away from the oyster farm, might be a kilometer away, and then they can find their way right back to it, even though the water is incredibly murky. So they're probably using sound. They can hear the oyster reef, and there might be other ways they can navigate, but they're really able to move around in this area. So this is, again, they have this really strong diurnal pattern where they sleep. There are other species of fish that are known to do this, basically hunker down and go to sleep. But this is one individual fish, an example going from September 1st to September 12th. And you can see the day-night cycle of this particular fish where he's moving about 100 meters per hour during the day, and then he's basically stationary at night, or she. We don't know if it's a he or a she. So this was pretty characteristic. The average fish was moving about 100 to 600 meters per hour. So again, for these relatively small fish, they're kind of constantly on the move. Some of them moved over a couple kilometers in an hour, the ones that were really moving fast. They had really strong fidelity to the oyster farm. They really liked the oyster farm. And again, they really appear to be navigating in this environment. So I'm wrapping things up. I guess to bring all this research back to some of the management implications for sheepshead, some of the things that we found that I think are relevant for sheepshead management. And of course, we identified these general areas where sheepshead are spawning, so that could offer some opportunities for the Division of Marine Fisheries to potentially monitor the sheepshead spawning aggregations in the sense where they now know where they can go and maybe estimate abundances of the spawning population or learn more about their reproduction or potentially protect them if need be. We found this really interesting discrepancy between the size of spawning fish and size at maturity. So what you're looking at here, these are the fish that we caught and confirmed that they were spawning around the Cape Lookout area. They were all really big. They were all over 40 centimeters. So these are really big sheepshead that we caught. And we didn't catch any small sheepshead, which we were pretty surprised at. Another study in South Carolina determined that most fish reach maturity and should be capable of spawning at a much smaller size. But for some reason, we didn't see any little fish in these spawning aggregations, which was kind of peculiar. And that also held true for all the other trawl data that we kind of scoured over. So if fish are in fact spawning at that size, we'd expect to see many more joining these aggregations. And we have a couple hypotheses for why this might be happening. Number one, recently some other studies have shown that some of these spawning aggregation species actually have an adolescence period, like a teenage years, if you will. So they might become reproductively mature, but they might not right away start spawning until they get a little bit older. So that's kind of one interesting hypothesis. Another one might be that the little ones are just spawning somewhere else. So that's something that needs to be figured out. And finally, and this is something that the Division of Marine Fisheries is trying to figure out right now with sheepshead. The harvest size limit is at 25 centimeters. I think that's 10 inches. And a lot of the fish that are being caught are immature. So there is some concern there. Even though the population seems to be doing well at the moment, there's increased interest in sheepshead, and the majority of the fish that are being caught are not sexually mature yet. So that could be an alarm bell. So we'll see how they deal with that. So that's it. I'll leave you with this little quiz, and hopefully at this point you're going to be really good at this, and I'm happy to answer any questions.
Do you have any data? Number of the stock, say from today back 20 years in our area?
Yes. So there's a question about the status of the stock and if there's data available going back to see how the health of the stock is. And there is a student at NC State has done some of the precursor work for a stock assessment. I think his work only went to around 2020, so not the last 5 years. And he put together, he used the data that are available on sheepshead, and his results suggested that the stock, at least as of 5 years ago, was doing pretty well. But the data availability of sheepshead is really sparse. Like none of the surveys really catch them all that well. Compared to some, other species of fish. Sheepshead just don't show up in a lot of the sampling gear very effectively. But sheepshead could be one of these species that are probably benefiting from climate change. They're kind of right here up at the northern limit. I think Chesapeake Bay is kind of the northernmost really big population. So increases in temperature could be benefiting sheepshead at the moment. But yeah, but hopefully in the next few years, DMF finishes a stock assessment and can give more information on the stock status. But I'm probably over answering this question, but for now, all the regulations are basically based on just landings and effort data, so which is not ideal. Any other questions? Yep.
Or the eating of the sheepshead, is it tasty? What is it like as compared to other fish?
So sheepshead are like a white fish, and oh yeah. How tasty are sheepshead? So I think there's probably a lot of people in the room that would say that sheepshead are delicious, white flaky flesh. When I was in grad school and we would catch them, we would do this Mediterranean dish where you gut them, stuff them with lemons, coat them in this salt paste crust and bake them whole, and then you break the salt with a hammer, and oh, it was like some of the best tasting fish I ever had. Any other questions? Yep.
I don't know anything about the acoustics and the tracking devices you use, but I was wondering, is there any chance that just the fact that those little fish are pinging changes how the prey fish go after them, or they interact with the oyster structure?
So the question is, if in these acoustic transmitter tags, they're making a noise, and does that kind of affect the whole ecology of the system, basically? So the ping is it's really high pitch, so probably right off the bat, some species aren't going to be able to hear it. There has been concern in these studies that those high pings might be detected by dolphins, and dolphins it might be sort of like a dinner bell for dolphins to come and eat them. In terms of sheepshead, and especially in the habitat where we did this, was most of the time the depth was like this, no dolphins going back in there. And sheepshead are eating a lot of invertebrates, so I'm guessing it probably, at least in our system, that probably wasn't a big factor. In other systems, it might be an important factor, though. Good question. Other questions?
Do the sheepsheads stay around the oyster farms that you studied, or at some point do they age out and leave and go on to other habitat?
We've caught big sheepshead. We've caught sheep. Repeating the question. So the question is, do sheepshead use oyster farms throughout their life cycle, basically? And yeah, we found sheepshead of all sizes on oyster farms. In fact, this video right here is, we call this a lift trap, and we use this to catch juvenile sheepshead. We would just leave these out in seagrass beds, come back once a month, get one person on either side of them, and then just pick them up, and then there'd be some juvenile sheepshead in there. Doing that, we've caught 9-pound sheepshead. They just, I think maybe they're sleeping in there or something. We pick them up, and we're just like, oh my God, like there's all these little fish, and then a 9-pound sheepshead just sitting like right in our face. And I think we've caught big sheepshead on oyster farms with that method. So they definitely like oyster farms still. Yep.
What do they do in the oyster farm?
So what are the sheepshead doing in the oyster farms? So I suspect they're nibbling all around the oyster farms. So they might be eating biofouling that accumulate on the lines and the pilings. But if you've been around oyster farms, or if you've looked closely at the oyster farm baskets, there's lots of like mud crabs and stuff crawling around on the outside of them. So I bet you they kind of swim around the oyster gear, trying to grab something that's crawling in the meshes of the oyster farm.
Jim, we have a question from the online audience. The question is, well, first they said, great talk. Are the largest sheepshead able to escape from dolphins or other predators that lead to large ones near Cape Lookout?
So first of all, there's no information on dolphins eating sheepshead. It could be possible. I've heard of dolphins eating pretty large red drums. So dolphins can eat some pretty impressively sized fish. But to my knowledge, there's no information available on dolphins preying on sheepshead. Again, I'm sure it happens. There's not a huge amount of data on dolphin diets. And the data we do get often come from sick individuals. So yeah, not enough information to answer that one.
I guess they have a follow-up here that says, do the small ones avoid the aggregation due to predation?
Oh, okay, so that's what they're getting at. Okay, that's definitely something we've thought of. So the smaller spawning fish, they might spawn in different locations simply because making the trip out there off the beach is just kind of hazardous for a fish of that size. So that's definitely possible. And so that's why we'd like to continue sampling the fish across the full size spectrum that time of year to see if we can find smaller fish using different habitats to spawn in. It's definitely possible. It's a good question.
Do other fish in the world have teeth that look like these?
Yes, so there are other species that have teeth like sheepshead. I mean, even red drum have some pretty impressive pharyngeal teeth. There's this freshwater species called the freshwater drum, which actually also has the local name Sheepshead, but they're not related, has pretty similar teeth. So it's not uncommon. I don't know what percentage of species, but it's probably pretty high. 40%, 50% of species probably have some type of teeth inside of their mouth. You might have to look a little bit more closely, at least of the fish that are a little bit more advanced, like the bony, spiny fish. But it's definitely very visible in sheepshead, especially because sheepshead, you can see their teeth. Just, you don't have to open their mouth. You can just see those big incisors staring at you. Any other questions? Yep.
On the very first graph you showed numbers of landings, it was really, really low in 98, 99. That didn't have anything to do with Dennis Ford, did it?
I don't know if there's been an analysis of that, so there's some low catch years really early on for sheepshead. It could be. I've read, and what kind of makes sense to me also is that sheepshead are kind of, to catch sheepshead requires kind of a unique approach. I'm not a good sheepshead fisherman. There's people in here that can catch sheepshead. But I think probably back in the late 90s, it was a little bit more of a niche knowledge base that actually knew how to catch sheepshead. And I think with the internet and all this online internet content, videos on how to do everything, including catch every single type of fish, I think that has really just kind of disseminated the knowledge base so people know how to catch sheepshead, what kind of bait to use, how to handle them when they're biting. So I think prior to the early 2000s, a lot of people might have just incidentally caught them, and probably wasn't as many people. But now people are learning how to do it, and they're going out and forced to go after sheepshead. Other questions?
I have a question. If the stock assessment plan, you got to do the stock assessment, how do you know to do the management plan? If it's so hard to do the stock assessment.
Well, I mean, if the stock assessment is conducted and then it's accepted as like it makes sense and it passes muster and peer review. I mean, they've certainly made fishery management plans of species that maybe are data poor, but yeah, I mean, that'll be a process that DMF will conduct. I'll be interested to see how it plays out, but I think they're definitely waiting to see if they can get a stock assessment approved, because then they can base the fishery management plan on what the stock assessment is showing. Otherwise, it's a lot harder.
I was going to say the flesh of the sheepshead, I believe, tastes better, say, compared to the black drum, because the black drum's eating fish and the sheepshead is eating shellfish.
Yeah.
So I think the flesh tastes a little bit like shellfish myself.
Yeah.
Yeah.
It probably does have something to do with what they're eating.
Yes, exactly.
And some of them, we've opened up, big adults, we've opened up their stomachs and it's just full of seagrass. It smells terrible. It's like a fermentation chamber in there, but there's like grass-fed sheepshead. I think Andrew, you ate that one, didn't you? Did it taste like grass-fed? Other questions? All right, well, I appreciate everybody coming out, and hope to see you again.